So I'd just like to start by thanking the organisers for inviting me to talk. Hopefully I'm going to take you somewhere a little bit different now. So about a year ago I joined the Oxford Biomedical Research Centre. That's a collaboration between the hospital in Oxford, the John Radcliffe, and the university. And I'm part of a broader group within clinical microbiology that's working towards replacing traditional culture-based microbiology, like these E-tests shown here. The antibiotic doubles in concentration each time you go up a gradation until you get to a certain amount which kills the bacteria. You see a kill zone. So that's a very kind of a way that Alexander Fleming would have been quite comfortable using that, and that's still standard practice. To actually go into an entirely genetics-based approach, we say, you know what, let's not do that. Let's just sequence the whole genome of the bug, look at the mutations, and work out, infer which drugs we can use to treat that infection. Now, we've had a bit of talking the last day or so about reproducibility, so I'm just going to change my title a little bit, and towards the second half of the talk, I'll show you some reproducibility analysis. So this sort of broad unit that I'm part of, Modbed Micro, we're on the top floor of the main building, John Radcliffe. Tim and Derek are infectious disease consultants and sort of started getting the ball rolling. Sarah's a statistician, Danny's a geneticist, Sam's a mathematician, although he's just moved to the EBI, and now me as well. So it's a very, very interdisciplinary group, and obviously there's a lot more people than I've shown here. So this is how it usually is for most bacterial infections in hospitals at the moment. The clinician will take a sample, it will be cultured, plated out in some way, perhaps automatically, and then a report returned to the doctor, an antibiogram saying it's resistant to this antibiotic, it's not resistant to this one, and then they make the decision. Most bacteria, this is fine. It may take a day or two, but that's quick enough. The exception are the mycobacteria like tuberculosis, which, because they grow so slowly, the culturing stage takes weeks, and so you're looking at somewhere, if you're lucky, getting a result back in 6 weeks. Now TB isn't you might be thinking, oh, TB, it's a bit Dickensian. TB is the single largest infectious disease killer worldwide at the moment. And there's also a lot of overlap with HIV/AIDS. So a lot of people with HIV/AIDS end up dying of TB as well. For example, you may not know, in the UK we actually have more cases in the UK than North America. So for a high-income country, we have quite a high prevalence of TB. So what we're trying to do as a whole, this is where I fit in, is replace this with genetic sequencing, which we can do because whole genome sequencing is dollars now for bacteria. Then we look at a panel of, say, 12 to 15 genes that we know are implicated in resistance, compare the mutations within a database to known phenotypes, predict the susceptibility. So how does this work in practice? If you've got, this is the database, you've got some genes, mutations, drugs, and then phenotype. So you take a sample, you sequence it, you look at the mutations, you look it up, you go, well, I've seen that one before, that's fine. I know it's resistant to this one, and all the others are sensitive. So presumably the clinician would choose one of these guys. So over time, we've built up a lot of whole genome sequence data. So we have about 10,000 TB sequences now, all from patient infections. And we also have staph sequences, C. diff, Klebsiella, all sorts of things. So this is just, don't pay too much attention to the detail, it just shows you all the TB mutations we've currently identified in the panel of genes. Now the great thing about this isn't fantasy. The UK switched over, so Public Health England switched over from the culture-based microbiology to this whole genome-based microbiology in March this year. So if you go to an NHS hospital now with suspected TB, you will get a result back in about a week because the TB will have had its genome sequenced. And that's happening at the 2 TB reference laboratories in Birmingham and London. And this is the first time this has happened anywhere in the world. And I think it shows how the UK can do these things quite well sometimes, because we actually have people in the group who basically can talk to themselves because they have roles within Public Health England, the NHS and the university, and we can move quite quickly on this. But we're not stopping there with TB, so about a year and a half ago we launched this project called Cryptic that's funded by the Gates Foundation, Wellcome and Newton. And the idea is to go out and collect about 100,000 genomes from about 12 partner labs across the world, so across all continents, apart from one. And the idea there is obviously to, by collecting these samples, sequencing their genomes and testing all the antibiotics, we can make this database much more comprehensive. Now where I come in is this is fundamentally an inferential approach in that it can only predict things that it's seen before. When it bumps into mutations it doesn't know, it's stuck. So I'm trying to come up with methods, and you can guess how, that will predict the phenotype for unknown or rare mutations. And these will become more common as we start using the second and third line TB drugs, because we just don't know, we just don't have the literature and work on the sorts of mutations that give rise to resistance to those that we do with the frontline drugs. Okay, we'll see if anyone's watching. So the proof of principle isn't TB, it's actually Staph aureus, which I hope you got from the title. And that was because we had this earlier study before I joined looking at whole genome sequencing in Staph aureus, and I chose DHFR because we've got a lot of crystal structures, it's quite well characterised, it's also quite small. Now, the drug we're looking at is trimethoprim, but the beauty of it is, because we've got all these clinical isolates, we not only have well-established resistance-conferring mutations, the 3 in red, well this one's actually a double, but we also have negative controls, because we have mutations that we see clinically that we know from experiment have no effect on the action of the antibiotic. So the prediction method now has quite a strong test, because it's got to be able to differentiate between these two. So it's really important to have negative controls, and I think this came up in the last session a bit. Now the way I'm doing that is, and this is not controversial to this audience, but it is a little bit controversial in microbiology, is to consider the effect of the mutation on the structure and chemistry of the protein. So I'm putting it in the structure and asking questions. So if we just zoom in on trimethoprim, this is F98, and this is trimethoprim. You can see, unsurprisingly, it's pretty close. And it works because it's a competitive inhibitor, so it binds in exactly the same place as the natural substrate, DHA. DHFR is on the folic acid pathway, so if you inhibit this enzyme, the bug dies. No surprises there. But what's really interesting is that most common resistance-conferring mutation is just an F to a Y. And this is actually quite characteristic in antibiotic resistance. So we just go from a hydrogen to a hydroxyl, and that's enough to confer resistance to the antibiotic. Actually, there was a poster out there as well looking at CAPG, which the most common resistance-conferring mutation for isoniazid is an S to a T, so you know, same sort of deal. But in a way, this makes sense, because when you think about it, evolution is solving a difficult problem. Because this is an essential protein, the mutation is abrogating the binding, presumably, of the antibiotic, but not affecting the binding of the natural substrate or the function of the enzyme. So that's quite a subtle problem. It's really easy to stuff up the action of a protein with a mutation, but it's difficult to balance those two things. And what it suggests is we should calculate how the binding free energy changes when we introduce a mutation. So this is delta delta G, except we're now perturbing the protein, not the drug. And if we see things bigger than 0, then we're going to call that resistance. I'm going to use alchemical methods. I've tried endpoint, it don't work. I didn't even bother doing docking. Just to say, we're following the best practice in doing decharging, van der Waals and recharging. Another thermodynamic cycle, you can see the decharging, recharging, decoupling going on here. We've got restraint legs in as well to numerically measure the cost of holding the restraint there. I just want to raise a brief point about Python. So it's been really useful using some of these tools developed by other groups, mainly in Python, because then I can start stitching things together and turning things more into a pipeline. I'm not there, and I've got some things in brackets that I want to try, but that's been very useful. So 10 years ago, I had nearly first publication in free energy calculations, and what we did was a fairly small number, and we were mostly kind of around n equals 1. And inevitably, at the time, we didn't do 5 repeats or so on because of the additional cost. But, you know, I think that time has passed now, so particularly for a protein of this size, let's just run repeats. It makes estimating the errors much easier. But I'm going to do something a little bit naughty, but in a way I don't mind, because all I'm trying to do here is I'm not trying to get an accurate binding free energy. I'm just interested in classifying that mutation as resistant or susceptible, because that's the way clinical microbiology works. It's essentially a qualitative field. So I'm going to run short lambdas. Okay, so these are only a quarter of a nanosecond long. I have run longer, but I haven't brought those results today. So that's the protein. Now, we don't have much ITC data. We do have some ITC data for the most common resistance-conferring mutation, F99Y. So this is 4 independent published experiments in the literature. And what we see is at 95% confidence interval, the error bars are just overlapping, so we're in reasonable agreement. Actually, if you run the lambdas for longer, this gets a bit better. For the other mutations, we just know from experiment whether they're resistant or not. Well, a bit more than that. We also know what's called their minimum inhibitory concentration. So that's the concentration of drug at which the bug no longer grows. If I wave my hands a bit, because trimethoprim is a competitive inhibitor, I can try and relate that down to the binding free energy. And then what I can do is there are tables of European standard breakpoints, they're called, for different species and drugs, and I can feed that in and then come up with 2 criteria. The first one's a bit simpler. It's just saying that the mutation is only going to affect the binding of the antibiotic, whereas the second one's a bit more subtle. It's saying, well, it can also affect the binding of the natural substrate. So we have 2 terms in here. Obviously, I have to run more calculations to do this one than I do this one. So if you just look at the first one, anything in that grey area we should classify as resistant. And what we see, pleasingly, is that all 3 resistance-conferring mutations would be classified as resistant. These, remember, are 10 repeats, but they're short lambdas. And of the 4 susceptibles, 2 would be classified as susceptible, and 2 would be classified as unknown because their error bars are overlapping with our threshold. So that's a pretty good start. Now, if we do exactly the same thing, but this time with the substrate in the pocket, what we see, broadly, we don't basically see any bars going to the right, or significantly to the right, which makes sense because that would be the mutation decreasing how well the substrate binds, which would have a fitness cost. But now that we've done both of these things, we can apply this criterion, which is the slightly more complex one compared to this one. But to do that, we have to put them on a 2x2, not a grid rather. So now everything in this triangle shaded region we would classify as resistant according to that criteria. And now, it should be, well, we get a slightly different picture. So we identify these 2 as resistant. That one's now classified as unknown because it's overlapping the line. These 3 would classify as susceptible, and this one would classify as unknown because it's overlapping the line. And in a way, when you think about it, maybe it's not too surprising that our F to Ls and L to Fs we're having problems with, because those are the ones with the largest number of atoms that we're perturbing. Okay, so we get, in theory, this should give us better discrimination. But is it reproducible? So all of this is n equals 10. So what I did was I just ran a whole load more free energy calculations. So we got 32 of each, and that's 32 of both with the drug and the natural substrate for all the mutations. And what we can do, and I'm not going to go through all of them because there's a lot here, really it depends where you fall on the grid. If V76A here, what you'd see if I showed you is that very quickly you home in on the fact that this is susceptible, and just adding repeats doesn't tell you anything different because it's far enough away from whichever criteria you want to use. The problem ones are going to be the ones that are near the line. So I'm going to look at those. Those are the more challenging cases. But first of all, let's just look at F99Y because that's a very important one. So that's the scatter of free energy calculations we get out of our 32 simulations. That's with the drug, that's with the substrate, and then that's a 2 to 98% kind of box and whiskers thing. The inset is the the variation in the estimated error. So you see some peaks and then it falls as you'd expect. If we then play bootstrapping games and just go through and keep picking 2, 3, 4, 5 samples with replacement and then saying, well, if I'd chosen these samples, what would the classification be? And then remembering that and then repeating, that will give us an idea of the sort of statistics of classification as we increase the number of repeats. And what we see is for the first criterion, where we're just considering the drug, after you get to about 7 or 8 repeats, we would always classify it as resistant. Whereas if we use the more complex criteria, remember we have to do more calculations here, after say 4 repeats, we would always classify it as resistant. And that's partly because it's a long way away from the threshold. So you're not picking the ones that are very low. If we look at this one though, we get a different picture. So if I just go back, it's a long way away from the first criteria, which is the vertical dashed line, but it's sitting on the second one. So unsurprisingly, this looks pretty good. It converges very quickly to saying, yeah, I would always classify this as resistant. But now, a lot of the time, we end up saying, well, I don't know, because the error bars would overlap with the threshold. So that's what I should report. And likewise for this one, now we end up saying it's unknown quite a lot, and just continually adding repeats here doesn't actually help at all, and here it's taking a long time. So perhaps inevitable. And then lastly, this one, the I83V. Again, what we see is that as we increase the number of repeats, we still end up with a lot of classified as unknown, and that's because we're very close to the vertical threshold, whereas here we very quickly, well not very quickly, but we do actually begin to converge to a result. Now I've got 1 or 2 slides left, so I'm quickly going to summarise. So hopefully I've shown you how free energy methods have the potential to be useful in clinical microbiology, which is perhaps something you might not have thought of. But what's been really vital for me is having access to these large clinical genomic data sets, because it gives me, for example, negative controls, it shows me where I should look, what I should try. And hopefully, and remember I'm trying to do something very specific here, I'm just trying to classify, that actually by running multiple really quite short free energy calculations, I can do a reasonable job of classifying resistance conferring mutations. As I said, I've got longer ones as well, and just didn't want to bring everything. Python's very helpful, I said that. And it just makes life much more straightforward, and it's easy to communicate as well when you're talking to colleagues and saying, well, I got my errors because I repeated it 5 times, and then they know what you're talking about. Now I thought I'd show this. This is interesting. So this is some of the conversation from earlier. So this is now allowing the simulations to be longer. And I can do this because I'm using this Python module Datrium to discover all the calculations, and it will just give me back all the ones that meet the criteria. So anyway, these ones, this is now the bin number times the duration. So what you can see here on this one is as you let the simulation duration grow longer, and this is the bootstrapped error, the error starts to increase, which I think we also saw on a poster outside. And presumably this is because you're moving away from that, everything's starting from they actually do start from different structures, by the way, but they're all from the same simulation. Everything's moving away, a bit further away from each other, so the errors start to apparently increase. And then at some point they would then start to decrease again. But it's interesting from a sort of philosophical point of view, what should I do if I'm trying to classify? Because actually, even though I know that my... But I've just done this quickly because I thought it would be interesting here, because it's not a complete picture. So for this particular mutation, remember these are the challenging ones because they involve a lot of atom changes. I do see this increase in the bootstrapped error as I push out the length of the lambdas, whereas for this one, I just see a continual fall. Maybe I will see an increase if I manage to go far enough here. This is a log scale, remember? So just thought I'd share that. So are my shorter sims actually really independent? Not sure, which would then mean my errors are probably underestimates. Okay, so I'll stop there. A lot of people have been involved with this on the clinical side. So Claire was the ID clinician who did most of the genomics work. Kevin at Brighton did all the e-tests. Angela gave me some data. And a quick sell, I've also got a citizen science project where you can classify the growth of TB on 96 well plates if you want to give it a go. We've just gone over 200,000 classifications. Yeah, and any questions, please. Thank you.
Thank you.